Now we're going to move on to how to separate these proteins or peptides if they're in a mixture. Common questions we as scientists or as people have when we have a peptide or a protein are things like, what is its sequence and its composition? That's a natural question if you want to learn about the protein. Just looking at a protein is not going to give you the sequence. We can't see the amino acids that is made of a protein. What does it look like three-dimensionally? And how does it fold? We also think about what is its role in an organism. If I isolate a protein, I want to know what it does. If I found casein for the first time from milk, I'd want to know, well, what is the importance of casein in milk? How is it regulated? This is a very important question when we're talking about looking at signaling in cells, for example. Being able to understand regulation of proteins can help us understand diseases. How does it interact with different things, other macromolecules, other proteins? Where is it found in a cell? Where is it localized? And generally, what are its kind of properties, its chemical and physical properties? To understand these peptides and proteins better, one thing we first usually have to do is separate it out so we can study it uniquely. Now, when you have an organism, it's filled with different peptides and proteins, and they're all mixed together. There's no easy way to particularly purify the protein that you are interested in. And so there are multiple types of purification methods. We can purify things using their amino acid sequence because they have different sequences of amino acids. And the types of amino acids that are in a protein can also give the protein a very general property. For example, if there are a lot of polar amino acids, the whole protein itself or the whole peptide itself will be very polar. If there are a lot of hydrophobic or non-polar amino acids, oppositely, the peptide or the protein would be very hydrophobic. Some polypeptides also will bind to specific targets, and we can use this to our advantage if we know that they bind to targets, and we can fish them out, essentially. We can give them a bait of something we know that they bind to and remove them from the mixture. Usually, we separate peptides and proteins based on their physical or chemical properties. In particular, we use the charge of the whole peptide or protein, the size, so its molecular weight, its affinity for some specific other molecule. This is what I was mentioning about fishing. If we know it binds to something, if we know it has an affinity for something, we can use that. How soluble it is, so we can put it in different solutions and it should be soluble in different solutions. We can use that to our advantage. How hydrophobic it is, and how stable it is thermally under different varieties of heat. One particular type of separation method is the most commonly used, and that is using chromatography. This is any type of separation in which the protein is able to remain fully folded and is separated based on one of these particular chemical properties. We've talked about chromatography in organic chemistry, and chromatography is also often used for small organic compounds, similarly to separate them from a mixture of other compounds. Just like in organic chemistry, we use a column usually to separate out our peptides or proteins. we take a mixture of proteins and add it to a solid phase, to a column solid phase, and use a particular liquid that helps to move and separate the proteins. Here, I want you to remember the properties of chromatography that we learned in organic chemistry. Proteins that are more alike to the solid phase will move slower. They'll stick to the solid phase. And proteins that are more alike to the liquid phase will move faster. They'll move with the liquid phase. Using this, we can separate out a mixture, and usually we get bands of different proteins that come off of our column at certain times. And by collecting each band and then testing it via other means, we can isolate our particular protein of interest. Now, there are different types of column chromatography, and they're going to use different types of phases for their column. One type of column chromatography is called ion exchange chromatography. This is a type of chromatography that separates the protein based on its charge. Here, we add the mixture to a column that contains cation exchangers. So this is going to be any type of resin. The column itself is packed with negatively charged ions. That means that anything that is positively charged is going to bind more so with the resin. And anything that is negatively charged will not bind with the resin, so it will go through the column faster. So again, positive charge goes slower, negative charge goes faster. Then, based on the size of the protein, we can distinguish it a little bit more. So things that are positive and large, they're big and bulky, will move through the column slower. They'll move the slowest, in fact. And then anything that is maybe a little bit smaller, but generally is still positively charged, is going to move still a bit slower. And again, those are because they're attracted to the solid phase. They have a positive charge, they want to stick to the column. Next, anything that's still kind of small, but it has a negative charge will move faster through the column. And the fastest will be things that are very large and heavy and have a negative charge. So here again is how things are going to move through the column. That is an ion exchange column. This is an example of an ion exchange column where we're using a resin with a negative charge. You can use a resin with a positive charge and still move proteins through based on their particular pH or charge. In this case, it would be flipped how they move through the column. Another type of column chromatography is called size exclusion chromatography. This separates the proteins based just solely on their size, on their molecular weight. Again, a protein mixture is added to a column, and in this case, the column has these cross-linked polymers. What happens is that the beads of the polymer itself are porous, and these beads, if you were to zoom in on the polymer, have these tiny, small tunnels in them. Those small tunnels entrap very small proteins, but the very large proteins do not get entrapped as much. So for a size exclusion chromatography system, very large proteins, proteins with a large molecular weight, will move very fast. They don't get stuck in the polymer. And very small proteins do get stuck, and so they move much slower through the column. Lastly, there is a type of column chromatography called a binding affinity or just affinity chromatography. In this case, the actual column itself is already loaded with a particular type of resin that has a special molecule attached to it, a molecule that the protein is known to bind. Sometimes we actually express proteins or make proteins that have a special segment on them that will bind to something that we know. That's so that we can separate these proteins using an affinity chromatography. But again, the general principle here is that you load the column with beads that have some type of special ligand of interest, some type of molecule of interest that the protein is known to bind. The protein will then bind these particular molecules, and all the other mixture will wash through. Well, now you have protein that is stuck on your column. It's actually stuck and bound to those ligands of interest. A second step of affinity chromatography is to add some type of solution that allows this binding to be disconnected, to cleave off the protein from the ligand. When we do this, it actually will wash the then purified protein, the one protein we were looking for, from the column. Now, another type of separation that we use to look at proteins is called electrophoresis. Electrophoresis is an analytical type of separation. Column chromatography does not tell us how much of a protein is there. It just allows us to separate our protein from other proteins that we don't want. So separate it away from a mixture. Electrophoresis actually tells us some information about the protein. What happens in electrophoresis is that an electric field pulls proteins through a gel according to their charge, or in some cases, according to their size. The gel matrix is going to hinder the mobility, so it'll change the mobility of the proteins based on their size and shape. One of the common gels that is used is called polyacrylamide, and it separates proteins via this polyacrylamide gel electrophoresis, which we abbreviate as PAGE. So polyacrylamide gel electrophoresis. A PAGE gel is usually what it's called. The setup for this type of experiment, this PAGE experiment of electrophoresis, is by loading different samples. These are different mixture samples of unknown content into our gel in these areas called wells. An electric field is then applied to the whole of the gel, and this current is going to allow the proteins to slowly move through. This is because they're induced by the electric field. The proteins, again, will move at a different speed based on properties of the particular protein. And so you can separate out different proteins within a mixture in this gel. Another type of gel is the SDS PAGE gel. The sodium dodecyl sulfate PAGE gel. This is where SDS, or sodium dodecyl sulfate, is added to the protein before it is put in a gel. Sodium dodecyl sulfate is a charged molecule, and it will unfold the protein. So there's nothing folded in it. It goes from being a folded clump to its just linear chain of amino acids. It also takes all of the amino acids and gives the whole protein a uniform negative charge. That means that the protein is no longer charged in different ways. So protein A and protein B, which might have had two different charges, now they're both negatively charged. This type of gel makes everything uniform except for its size. And so the rate that a protein will move through the gel is only dependent on its molecular weight, its size. Here's an example of an SDS page and how you might use it. Here we can see this is each of the wells that has then been run. In these beginning wells, you can see there are multiple bands. Each of these bands represents a particular protein. It has separated into its individual area. This initially came from a mixture at the top and was separated into all these individual bands. What this tells us is that this particular sample has a lot of proteins in it. It has a variety mixture of proteins. And as we go from sample to sample to sample to sample, you can see that this last sample only has one band, one protein available. This experiment was actually taking some mix of proteins from cells, and they were using types of chromatography to try to separate out one particular protein. And you can tell using gel electrophoresis that they, in fact, did separate one particular protein. You can also tell what the molecular weight of this particular protein is. Again, using SDS page and this type of electrophoresis, you can calculate the molecular weight of a particular unknown protein based on where its band is on the gel compared to something we call a ladder. A ladder is just a certain sample mixture with known amino acids or known peptides and proteins in that mixture. Each band has a known molecular weight. So we already know the size of band 1, 2, 3, etc., and its precise molecular weight. These are standards. They are things that we already know. We can then compare our band from our unknown sample to the ladder, the set of known molecular weights, and we can get our actual molecular weight of the unknown protein. So this is how electrophoresis and SDS page in particular can be used analytically. It can be used to calculate something about a protein. Another type of experiment that can be done that helps calculate analytically information about a protein is using absorbance. We noted in the first lecture that the aromatic amino acids all absorb light in the UV region. Now, most proteins typically have some amount of one of these amino acids. Proteins usually have hundreds of amino acids, and it's typical that at least one of those amino acids will be an aromatic amino acid. So we can use how much that UV absorbs to calculate how much protein we have. So any protein and peptide with a known extinction coefficient, we can calculate its concentration. So using our Beer-Lambert law, we can measure the absorbance of a protein that we've collected, some unknown protein. We can use its extinction coefficient. This is a standard. This extinction coefficient is a standard that you would look up for a particular protein. And we know the L, or length of the cell that we are measuring our sample in. And using this, we can calculate how much protein concentration we have of that particular protein. So we're using some absorbance that comes from an aromatic amino acid, and we're using a standard extinction coefficient to calculate concentration. Now, based on what we've talked about, proteins in these complex mixtures often require more than one type of purification. So we don't usually just do a size exclusion chromatography or just do an affinity chromatography. We usually do a series of different types of purifications to then really isolate out our particular protein. During purification, determination of the location of the protein of interest can be performed by tracking its behavior. What I mean by this is you can make sure that as you are isolating and doing these experiments, you can make sure that you still have that protein there by checking how active it is. So for example, if I have a protein that makes lactate, then I can check from my solution that I have how much lactate is being made from that solution. That will kind of give me an idea of how much of the protein is there. So if a protein has a specific function, like if it binds insulin, then the fraction of that solution that binds insulin after each separation step is what contains my protein of interest. So generally, the function of a protein is called its activity. If I have a solution and I say that it is so much % active, that tells me generally also how much protein is there. I know how active, how much of that solution is doing the activity of interest, is able to convert A to B, for example. Again, this is called the protein's activity. The ratio of the activity to the protein concentration is called its specific activity. And I'll give you an idea pictorially of what that means. So again, if I have beaker A and beaker B, and they have my protein of interest in them, and they might also have some other protein in there. So they still might be slightly mixed. In beaker A and in beaker B, my activity is the same. So if my protein of interest is the blue protein, we'll say 2, 3, 4, 5, 6, 7, 1, 2, 3, 4, 5, 6. Oops. Counted one that was not there. 6 and 6. So you can see there's the same amount of protein in beaker A as there is beaker B, the same amount of blue protein. Their activity should be the same. Their ability to do some type of conversion or whatever it is that they can do will be the same in beaker A and beaker B because there's the same number of protein. But beaker B is more concentrated with the protein of interest, which is the blue protein. That's because there's less other stuff around. So beaker B has a higher specific activity, meaning that per everything else in the beaker, there is more of the protein of interest, more of the protein, the blue protein. So these are what these words activity versus specific activity generally mean. Lastly, we're going to talk about how we figure out the sequence of these proteins. This is essential in general for biochemical activity. And if you look up any protein on the PDP, the Protein Data Bank, then you will see its amino acid sequence. Generally, we actually determine the sequence of an amino acid string in a protein from its DNA sequence. But there are other methods where we can take the protein and break it down and individually figure out which amino acid goes in what order of the chain. One of those methods is called Edmond degradation. And generally, you're going to selectively do N-terminal modification, then cleavage, and then identification of each amino acid. The more used type of sequencing is called mass spectrometry sequencing. This uses a mass spectrometer. This is an analytical machine which can measure mass, and it will precisely identify the mass of a peptide. And because it is so precise in its identification of the mass, we can then know its sequence. Here is just a pictorial example of how protein sequencing can be done using mass spectrometry. Essentially, what you do is add in your whole peptide sequence, and this whole peptide sequence will then be individually broken into chunks, and it will be broken each time after each amino acid. Essentially, we can then use this one by one list of amino acids to then put together our chain of our peptide. We can also measure the entire protein and understand its whole mass. This image here shows what the mass of a whole protein looks like. Mass spectrometry and protein sequencing using mass spectrometry is something within my field of research, using mass spectrometry to understand biochemistry. Okay, so that ends it for chapter 3. To summarize, from both this video and our previous video, we talked about the biological functions of peptides and proteins, the structures and names of those amino acids that are found in proteins, and we talked about ionization properties for both an amino acid or a peptide chain. And lastly, we touched on some of the methods that are used for separation and analysis of peptides and proteins.